So good afternoon, everyone. I'm Lauren Dunn. I'm the medical and health producer for NBC News. I'm really excited to be here today moderating this panel because we're talking about an issue that really impacts every single one of us, right? We're talking about aging, and no matter who you are, where you're from, your race, your ethnicity, everybody ages. And I think most of us could also really agree that we don't like it, right? That, aging isn't necessarily fun. And I think that's why I think for as long as we can remember, people have been on this quest for immortality, right? This idea to halt our bodies from growing old. And now it really turns out that we're actually closer than we've ever been before to halting the aging process. And I'm not talking about vanity, right? That was the first thing I thought of. We're talking about reducing wrinkles and all that stuff. But again, I think many people would like that. So if there's something you guys can do, I'd be happy. But we're actually talking about keeping our bodies younger, so our cells, our organs, because that's really going to help prevent a lot of the diseases that come along with aging. So think about arthritis, Alzheimer's, cardiovascular disease, even some cancers, right? These are all the things that start to happen to our bodies as we get older. And I think the need for this is more important than ever, because there's been these amazing advances in medicine and science, and we are living longer, but we're not necessarily living better. And these are the guys who are going to hopefully help us to live a little better, to increase this idea, our health span, instead of the lifespan, or maybe a little bit of both. So I want to introduce everyone today. We have Matt Kaeberlein from the University of Washington. And thanks for switching the slide for me. We have Saul Villeda from University of California, San Francisco, and Ned David from Unity Biotechnology. He's going to kick us off today, so why don't you get started?
Awesome. Well, thank you, Lauren. It's a total honor to work on aging biology at this moment in time. And the reason this moment in time is special is we're beginning to witness the intersection between understanding the fundamental mechanistic elements that drive this thing that we call aging. We're seeing the intersection of that with the ability to make drugs that impact those mechanisms. And so the world is going to change in the coming decades, and the way we're going to wind up thinking about how we organize our lives, when we retire, what's the nature of youth, what's the nature of being old, is beginning to shift. And this whole story I want to talk about today, which I'm going to try to tell to set context for where does our work fit, where does Saul's work fit, where does Matt's work fit, really begins with this woman. That is Jean-Louis Calment. She was a French woman, a resident of Arles, and she was very much a French woman. She rode her bicycle every day until she was 100 years old. She smoked cigarettes until she was about 117. Toward the end of her life, she got really into chocolate, and so she was eating 2 kilograms of chocolate a week. Just get your head around that. And she only wound up being put in assisted living when she somehow managed to catch her kitchen on fire. Now, this picture, taken a year before her death at 122, Jean was the oldest living human being ever. Now, recent demography, which is sort of the study of populations, suggests, suggests that the 150 years we've had of ever-increasing human lifespan, this number and its rate of increase is gradually ground to a halt. And what we're beginning to witness is this maximum achievable human lifespan. Now, which is, by the way, somewhere between 120 and 130. Now, we did not come here today to talk about natural human lifespan. We wanted to talk about something a little more fun, which is augmented human lifespan, which is what becomes possible if you are willing to disturb the universe of naturally occurring human biology. Now, if you guys are awakened at 2 in the morning, in the middle of the night, say 2 weeks from now, I hope there are 3 ideas that you will remember from this talk. And the first is that aging is not a rigid thing that is unchangeable. It is a flexible, malleable thing, and nature has bent and twisted rates of aging in creatures throughout evolution. Now, to do this, nature has created a series of molecular control knobs that it twists and presses to change the duration of an animal's lifespan and health span. And we as scientists and drug discovery explorers, we are learning how to turn these knobs safely. Now, why do I believe all of this is true? Well, let's look at the flexibility of aging. So if you look across evolutionary history and phylogenetic space, look just in the class of mammals of which we are all members. The shortest living mammal is the tiny shrew. It's this cute little guy over here, lives less than a year. The oldest living mammal lives over 200 years. It's the Bowhead whale. People actually don't know how long this creature can live. It's at least 200 years. And these are creatures with more or less identical biochemistry, with greater than a 200-fold difference in lifespan. Now let's look at the order of rodents. I think most of us are familiar with the field mouse. This creature, if you feed it and treat it nice, will live to about 3 years. But its closely related relative, the naked mole rat, can live over 10 times longer. So if you look at another relative, Brandt's bat, it lives 12 times longer. And it's, again, these are all tightly clustered creatures in evolutionary space. Let's go further afield all the way over to the phylum of mollusks. So the hard clam, it's something you guys are familiar with if you've gone to a clam bake. Left undisturbed and uneaten, it will live to about 40 years. It's very visually similar and closely related relative, the deep ocean quahog. No one knows how long this can live. The oldest samples that have been found are over 500 years old. For all we know, this could live 1,000 years. And these are creatures that, again, are tightly clustered in evolutionary space. So I think this suggests, obviously, that aging is this flexible, malleable thing, and nature, for reasons that no one told us about, has been bending and twisting the aging process. Now, why do we think aging has molecular control knobs? And why do we think these knobs can be turned? Well, the first turning of an aging knob by people was by accident. So in the Great Depression, one of the things that happened is people had very little money. And so there was an investigator named Clive McKay who had a giant colony of aging rats. And he wasn't studying aging, but he didn't have enough money to keep the rats fed. And so one of the groups, he just fed them every other day because he didn't like that group as much. And the other group he fed every day. And something ironic happened. The ones that he didn't feed as much lived 30% longer. And it was the first time there was a systematic, scientific, accidental exploration of how you can alter lifespan. Essentially, restricting calories can increase the lifespan of a creature related to us by over 30%. Then there's this molecule, rapamycin. And Matt's going to talk about rapamycin later. This was a molecule discovered in the early 70s on Easter Island. And this is a molecule that if you feed it to rodents and other creatures, which Matt will talk about, you can extend the lifespan of a rodent by over 30%. Now, this is, in fact, what it looks like. These are two rodents sewed together at the hip, and their circulatory systems are fused. And so one is young, one is old. So the blood of the young is flowing into the old, and the blood of the old is flowing into the young. And this technique is called parabiosis. And what it happens is that you can extend the lifespan of the old one by as much as 15% by perfusing it with the blood of the young. And Saul is going to talk about where this work leads us. Now, what I want to talk to you guys about is something very cool that we've been working at Unity for the last 5 years. And to sort of explain where this work fits, I have to explain a little bit about how we think about the mechanisms of aging. So if you think about aging, we do not believe there's a single reason it happens. We think there's a overlapping set of mechanisms that contribute to the thing we call aging. So you guys may know what stem cells are. It's clear that these are lost as we age. It's thought that contributes to tissue dysfunction. There's direct metabolic control of aging, and that's what Matt's going to talk about. You can see that in calorie restriction. You can see it in rapamycin treatment. Then there's this loss of circulating youth factors that Saul is going to describe. Now, there's this big question mark. This is where all the future stuff, where we think, the actual ticking clock of aging resides. Now, why do we even think there's a ticking clock? We think that because there are pieces in all of us, our germlines. That's our reproductive organs that then become the reproductive organs of our children and our children's children. These are immortal. And there's some mechanism that protects them and makes them special. And understanding the difference between that and the rest of us is what's inside that circle with the question mark. Then there's this mechanism called cellular senescence. We've been working on this for the last 5 years, and we work on it not because it's the only reason you age. It's not. We know this. We work on it because it's very druggable. And the basic story is this. This is you at conception. You begin as a single cell. You divide a bunch of times over the arc of your life, as many as 50 times. And as cells approach 50 cell divisions, they will encounter some form of stress. And when they encounter this stress, they pull an emergency brake and they stop dividing forever. And cells that pull this emergency brake, we call them senescent cells. Now, this is a super important emergency brake. You do not want to mess with it. If you do mess with it genetically, mice are born normally but wind up full of tumors before reaching reproductive age. So don't mess with the e-brake. However, as we age, these senescent cells begin to accumulate in our bodies. So my son, who's 8, has no detectable senescent cells, whereas my stepfather, who died at 87 from Alzheimer's, was full of these cells. Now, before our work, and that of our collaborators, both at the Mayo Clinic and the Buck Institute, is no one knew if these accumulated senescent cells were good for you, bad for you, or neither. And so what we did collaboratively with Jan van Deurzen and Judy Campisi is we genetically engineered mice where we could eliminate these senescent cells whenever we wanted. And for the first time, we got to ask, what happens? Well, this is what happens. These mice are siblings, born within seconds of each other from the same mom, and they're more or less genetically identical. And to give you a sense of their age, these mice are about 70 years old, assuming you were a mouse. This guy on the left is blind, osteoporotic, and frail, has kidney dysfunction and cardiac dysfunction. This one on the right lives 35% longer, but that's the boring part. The cool part is it has a profoundly extended period of time we call healthspan. This is the period of time this creature lives free of many of the chronic features of aging. Things like kidney dysfunction, cardiac dysfunction, frailty. And what we're doing at Unity is we're making drugs that do that. But I want to show you a few cool things that happen when we eliminate these cells. So this is a CT scan of mice with and without senescent cells, so you can see their skeletons. And if you look closely at their skeletons and you look at the femur, this is the bone in your leg, it's the longest and strongest bone in your body, and you look down the central axis of the femur, what you see is this 25% thickening in the wall of the bone. And this is in mice that are equivalent to an 80-year-old person. And if you look at the animal that has the senescent cells and look inside the bone, it's all pitted out like the surface of the moon. So it suggests that these cells are somehow actually dissolving bone. Let's look at spines. So one of the great indignities of aging is back pain and the gradual loss of the intervertebral discs. Well, when we eliminate senescent cells in mice over the arc of their lives, we can preserve 41% better the intervertebral disc volume. And I'm someone who suffers from degenerating disc disease. It runs in my family. So every morning when I wake up and there's some kind of weird back pain, it's highly motivating because I know the phenotype we can see in mice, and I believe that we're going to be able to achieve this not just for my children, but for myself. So it's highly motivating observation. So obviously these animals look different, but one of the fun things to think about is, what do they do that's different? So we did a cool experiment. We put these guys in a box, and we put a camera on top of the box, and we record what the mouse does for 15 minutes. And if you've never seen an experiment like this, this is what the data looks like. So see that crazy-looking pink line? So that's a young mouse running around for 15 minutes. Now, what do you think an old mouse is going to do? Well, it's that. Kind of a bummer, huh? Okay, so and that's only a mouse that's only 6 months older. Okay, and so as you can see, it runs around a lot less, and for reasons that the mouse is unwilling to share with me, it hates the center of the cage. Okay, now, so what we did is we eliminated senescent cells during its life, and we compared what happens. And what you see is that as you go from early life to midlife, the animals, if you eliminate senescent cells, run around threefold more. Basically, an 18-month-old animal runs around like a 12-month-old animal, which is awesome. But what I think is even neater, they go to the center of the cage like a young animal. Now, if you were to go to the FDA and you were to show them this data, they would kind of look at you like you're totally weird, because there will never be a drug that has a label that says, I want to go camping and take up a musical instrument, okay? But I think this tells you about the power of the biology that we're perturbing, and that's why I think it's very cool. Anyway, stay tuned, because over the next few years, we're going to be putting these drugs into people. Our first drug will be in human beings for osteoarthritis of the knee in the next 12 to 18 months. So we're keeping our fingers crossed. All right, thank you. By the way, be sure only hit the right one.
Sounds good. All right. I will follow Ned, and I will stand as well. Great. So, hi, everybody. Again, my name is Saul, and I'm an assistant professor at the University of California, San Francisco. Now, I'm going to start with this image, and I really like it because it sort of shows that aging is both an active process that everyone, it's just something we're all going to have to go through. And the other thing, it shows that it's a continuum. The one thing I will mention, just following up on what Lauren said, is my birthday was last month, and I'm going to admit that ever since I went past 30, I keep spending a little bit more time in the mirror looking for the wrinkles and the gray hair, and heaven forbid, the loss of hair. That to me is just frightening. However, we're interested in a lot more than just these physical hallmarks, right? But more than just the gray hair, the wrinkles. I'm at least really interested in higher order cognitive functions. How about those things that you can't see, but that are really important to us, very intimate to us, which is sort of our cognition. You know, it makes us who we are, and it allows us to think the way we think, and it allows us to learn. And that's also impacted by aging. So I'm interested in what's driving that. Now, in my lab, what we're asking is actually, can the aging process be reversed? So not just can we extend it, not can we play with it, but really, once we're already old, can we somehow reverse that? And in particular, can that apply to the brain? Now, why is this important? Well, as we get older, we suffer from age-related cognitive decline. So I like to start this graph. You'll notice it goes from 30 all the way to skull and crossbones. So I like to start at 30 because I'm actually quite upset at what happens before 30, which is an increasing slope. So you're actually improving if you're less than 30. We get at 30, and we're doing great. It's awesome. And then we see that it starts decreasing. And there's this magical moment which actually correlates really well with retirement age, at which point that decline actually sort of causes susceptibility and vulnerability to neurodegenerative diseases, things like Alzheimer's disease. So what's interesting about that is even people that have genetic predispositions for things like Alzheimer's, when they're young, they're not actively suffering from that cognitive decline. And why is that? You know, that to me suggests that there's something about that youthful state, something about that young brain that somehow naturally knows how to counteract that, how to sort of, combat the effects of aging. So the idea here is if we take someone at an older age that maybe has some cognitive impairments already, are there ways that we can bring them back? Are there ways that we can restore that youthful state in the brain to really prevent this neurodegenerative disease from beginning in the first place? And that's the motivation behind what my lab does. Now, in terms of cognitive decline, let me put this into everyday context for you guys. So I live in San Francisco, which means that every person and their mother has an electric car. In particular, Teslas seem to be very popular right now in the Bay Area. I'm an assistant professor, so there's no way that I can actually afford one of these cars. But follow me here. Let's hypothetically pretend that I can afford this car. And I'm going to drive to work, which is right atop a hill in San Francisco. And I'm going to park my car. But I have to remember, at the end of the day, no matter how frustrated or happy I may be with the data that's coming or the grants that I'm getting, I have to remember why I parked my car in the first place, because I want to go back home. So what's going to happen is my brain is actually going to notice these spatial cues that are surrounding my car. It's just going to notice it. In particular, I have a great view of the Golden Gate Bridge. Let's see, perhaps there's a rainbow flag nearby, and like food trucks are really popular in the Bay Area. I'm not sure if it's the same where you are, but for us it is. And let's say that my brain is always going to know where my favorite taco truck is. So I'm going to go to work, I'm going to come back, and my brain is going to use spatial learning and memory, and it's going to guide me to my car based on those spatial cues. I'd like to point out that if I could actually afford that hypothetical Tesla, it would be red so that you could see that I have a Tesla. However, again, my brain's going to use those visual cues. It's going to direct me towards my red Tesla. When you're young, you have no problem. This is great. However, what happens when you get older? Well, I'm sure all of you have been in a parking lot where you see that person with their car alarm and they're actually trying to use that sound to find their car. That is no longer spatial learning and memory. That is cognitive decline. That is your brain on aging. So again, this is sort of an everyday sort of example of the type of cognitive decline that we have that eventually is a sign, okay, there's something going on in your brain that aging is affecting. Eventually, we're going to have vulnerability to these neurodegenerative diseases. Now, what's interesting is this same type of age-related cognitive decline, it's actually conserved across species. So for example, in my lab, similar to me finding my red Tesla after a day of work, my lab actually trains rodents, mice, to find a hidden platform in a cloudy pool of water. So just like I'm using the Golden Gate Bridge, a rainbow flag, and a taco truck, they're going to use cues around this pool of water. They hate cold water, and it has to be cold, so they want to get out of there as fast as possible. So what we're going to do is we're just going to train them, and every time we put them in there, they get better and better and better at knowing, okay, I see that. That hidden platform is next to the red triangle, far away from the yellow square. And a young animal is able to learn this really well. However, an old animal can eventually find the platform, just like eventually you will find your car, but it takes them much longer, and they have to go through a lot more errors than a young animal would do. Now, I want to point out that just like Ned mentioned, old animals have changes in the way that they can move. So the reason that we put them in a cold pool of water is to sort of circumvent that. All of a sudden, we're forcing them to do this task. So we're actually just testing their ability to remember where this is, learning and memory. Now, using these really basic behavioral tasks, we can actually start to ask, number one, what is driving aging? Are there things that we find in an old animal that could promote cognitive decline in a young animal? What about the opposite? Can we then reverse cognitive impairments? If we take a normal aging mouse, can we bring it back? Now, in thinking about aging and thinking about this ability of promoting aging or reversing aging, what we call rejuvenation, we really took a step back and we thought, okay, it's not like the brain is by itself, right? The brain is part of the entire organism. So let's think of aging as a systemic process that's affecting every single tissue in our body. And we thought, what connects every tissue in our body? What connects the liver and the pancreas and the brain? Well, it turns out what connects it is blood. Blood flows through every tissue in our body. It's really almost like a highway where one communicates to the other. So we thought, can we learn something about aging and potentially rejuvenation by investigating blood? Now, how are we going to start asking these questions? Well, a really good way would be if we could mix blood. If we could somehow put old blood into a young animal and young blood into an old animal and old blood into a young, yeah, you guys get it. So what we do is we use this model known as Parabiosis, and Ned showed you the kind of creepy picture of it. I'm going to use cartoons because the creepy pictures kind of freak me out. So here we have in brown a young animal, in gray an old animal. And what we're showing is that by just connecting them, almost like Siamese twins, we can actually mix young and old blood, such that two hearts are now pumping one bloodstream. And we can ask two questions. We can ask, how is old blood affecting the young? How is young blood affecting the old? Really, we can start asking about pro-aging and pro-youthful mechanisms. Now, it turns out that work over the last, almost over 10 years now has really shown us that a few different things can happen. If you expose a young animal to old blood, it causes premature aging. They show signs of aging throughout different tissues. However, the opposite is also true. If you give young blood to an old animal, you can actually reverse a lot of hallmarks of aging, including in the brain. And we thought, okay, this really gives us two different strategies to potentially reverse aging. One is targeting things that are actively promoting aging, what I call pro-aging factors in old blood. And if we target them, can we reverse aging? And the other approach is finding youthful factors in young blood that we bring back. Maybe there's factors that as we get older just start diminishing or disappearing. Can we bring that back? Now, I'm going to give you a couple of sort of mini things that we've actually worked on or are actively working on. The first one is looking at these pro-aging factors. And what we did is we actually surveyed old blood. Targetedly, we knew, for example, that aging is associated a lot with inflammation. So we asked, okay, as we get older, how are inflammatory sort of factors changing with age? And we found that a lot of them are increasing. And we identified certain, almost like master inflammatory regulators that are promoting aging. And it turns out that using genetic models, similar to the ones that Ned was talking about with senescence, if we can target individual molecules in a young animal, you can actually prevent some of this cognitive decline. So rather than that animal having a hard time finding that platform, has a much easier time finding the platform. Not quite as good as a really young animal, but much, much better. And just so you know, in our lab, we study young mice that are equivalent to sort of the age of about a 25-year-old human, and we're studying aged mice that would be equivalent to about 65 to 70 years. So that's sort of the time frame that we're thinking about it. So to us, this really starts indicating that, yes, we can actually start targeting individual factors in old blood as a way to prevent age-related decline. Again, that's prevent. We're actively working on, can we actually reverse by targeting these old factors? We have some really interesting data that I think we will be able to, but I think it'll be a combination of factors that we have to target. Okay, so this is pro-aging, but what about the converse? What about the young factors? Can we actually increase or reintroduce those? Now, for this, again, a lot of labs throughout the world are still actively trying to find candidates. There's a couple of them out there that we're pretty excited about. The consensus is that we're going to have to find a lot of different ones, or at least a number of different ones to get full rejuvenation. But at least we do know that young blood itself can do something. Now, what we did in collaboration with my old advisor when I was getting my doctoral degree at Stanford University, we did the Parabiosis experiment. And absolutely, we found that young blood can increase stem cells. It can actually change your neurons themselves, the structure of your neurons. These cells that have been with you your entire life can actually reverse in their structure and look much younger just with young blood. We've done all sorts of different experiments. We've looked at inflammation, and that changes. However, none of us want to be hooked up to another person next to us, right? This is great, I'm the mad scientist, but this isn't what's going to get into a clinic. So we came up with an alternative approach, which is just injecting plasma. Just the factors in blood is actually enough to recapitulate a lot of the same things we saw in this Parabiosis model. Now let's think about that for a second. You're already elderly. You can introduce blood or young factors that you had when you were younger and somehow reverse the signs of aging, at least at a cognitive level. Now, what's really cool is that over the last 10 years, this isn't just a brain thing. This is actually throughout our bodies, and people around the world are seeing this. We've now seen it that it can improve muscle repair after injury. It can actually improve bone and bone mass, which goes down with aging. It's been looked at in pancreas and liver. All throughout the body, we keep finding that just reintroducing something into the bloodstream is enough to reverse these hallmarks of aging. So again, there's a lot of active investigations. Actually, Ned and I are working together on actively trying to find things that individually can be reintroduced into your bloodstream to sort of mimic the effects of this young blood. However, what I want to sort of make sure that you take home from this is this idea that aging is not final. Aging is not concrete, but rather it's very malleable. Similar to all of the evolutionary data that we have out there with animals living so much longer than each other, no matter how close they are with respect to evolution, there's other data that we now have where we can actually manipulate organisms and actually reverse the hallmarks of aging. It's really, really sort of adding to this body of evidence that we don't have to stay the way that we are when we're old. We can actually return functionally and physically to a younger state. Now, in our case, again, we're really excited about identifying both pro-aging and pro-youthful factors, and I think that this is one approach that we can take to try and reverse aging. As you heard, caloric restriction is another one. We're going to hear a little bit about one actual molecule that is already showing really great promise. But again, what I want you to take out of this is the fact that just because you're looking at yourself in the mirror and you start seeing the gray hair and the wrinkles doesn't mean that has to be the end of it. Scientists and biomedical research is really moving forward at a very fast pace to try and really investigate aging, understand aging, and even manipulate aging. Great.
Ned, put it back on play. So I want to say that after Saul's talk, I'm really sad because, no joke, I spent half an hour looking for my rental car last night after leaving this building. Yeah, I got it. Thanks. All right, so I want to just briefly come back to this concept that Ned presented about how aging is really a biological process, and one implication of that is that it's modifiable, and we can slow it down or we can speed it up. And this is a really sort of a profound way of thinking about health, because the biomedical enterprise for many, many years has really been focused on disease and waiting until people are sick and then trying to treat their individual disease. What kind of gets lost sometimes is the fact that essentially all of the major diseases, causes of disability, morbidity, and mortality in developed countries have at their root a single common risk factor. And it's not how much you eat, whether you exercise, how much you smoke, how much you drink, it's how old you are. Now, I know this is South by Southwest. I'm not necessarily advocating that you go out and engage in risky lifestyle habits, but I just want to maybe just a little bit. Moderation is the key, right? But I just want to make the point that aging is really what drives our susceptibility to getting sick and dying. And so what that means is if we can understand the aging process at a molecular level, which we are beginning to, and we can intervene in that process, we have the potential to really slow down aging and then at the same time prevent the onset and progression of all of these diseases simultaneously and before you get sick. So expanding this period of healthy longevity, not just how long we live. And I really think that's the take-home message of this session. So what I want to talk about is a drug that actually seems to do this, at least in laboratory animals, and that's this drug rapamycin. As Ned mentioned, it was found on Easter Island, or Rapa Nui. That's where it gets its name. It's been FDA approved for many years now. It's used primarily to prevent organ transplant rejection in people, and that pill there is actually the generic version of rapamycin. So this is now a generic drug that it can be prescribed off-label. So its clinical use is primarily to prevent organ transplant rejection, some forms of cancer. In the geroscience field, or the biology of aging, what we've seen is this drug really seems, across a wide evolutionary distance, all the way from yeast and nematode worms up to small mammals like mice, it seems to slow the aging process, robustly increases lifespan, and appears to reverse essentially all of the declines in function and diseases that go along with aging. So all of those age-related diseases that I showed there are either mouse models, or this is part of the normal aging process in mice, that there's evidence that rapamycin can slow down. And in addition to that, there's a variety of other disorders that may or may not be directly associated with aging that rapamycin seems to improve. So it really does seem to have quite a potent effect on somehow impacting whatever is causing our bodies to become more susceptible to these diseases as we get older. One of the interesting things that's emerged from the literature over the last 5 years or so is that in some cases, rapamycin doesn't just seem to slow down the aging process, it actually seems to push it back, to functionally make organs or even whole animals as if they were more youthful than when they started. And this is something that I personally was kind of shocked by, because I had never really put credit to the idea that rejuvenation per se would be that easy. So I'm not saying that rapamycin is going to take an 80-year-old and make them into a 20-year-old, but at least for certain functional measures, it does seem to set back the clock. And so that's been shown for immune function in both mice, and a little bit of evidence in people that short-term treatments with rapamycin can rejuvenate the aged immune system, help it respond to a vaccine better. It's been seen for cardiac function in mice, and this is the images there show some unpublished data from my lab, even for bone loss associated with periodontal disease. Turns out that somewhere around 65 to 70% of people over the age of 65 can have periodontal disease. So this is certainly an age-related disease. And short-term treatment with rapamycin in mice rejuvenates the alveolar bone that is lost during periodontal disease. So one of the things that my lab has shown recently is that even a short-term treatment with this drug can have pretty profound effects on lifespan. So that's what the chart here is showing. You don't really have to worry a whole lot about the data, but it's the percent of animals that are alive as a function of age. So these are old mice. And what we did is we treated them for 90 days with the drug. So they were treated from the time they were 20 months old to 23 months old. That's about the equivalent of 60 to 65 years in a person. And then we stopped the treatment, and we just asked what happens. And so anything that's to the right is an increase in lifespan. And what we saw was in this particular cohort of mice, 90 days of treatment with the drug after we stopped the treatment was enough to increase lifespan by 140 days. So the increase in lifespan was actually bigger than the length of time that they were treated with the drug, which is a little bit mind-blowing, at least to me. So that suggests that we can get profound improvements in health span and lifespan from a short-term treatment with rapamycin, at least in mice. So what we're starting to do now is to try to step out of the basic science realm and move into the clinic, in this case the veterinary clinic, to not only learn as much as we can about the aging process in pet dogs, but also extend the healthy period of life and hopefully lifespan as well in pet dogs. And so this project is called the Dog Aging Project, and the goal really is twofold. One is to learn as much as we can about aging, because we're going to learn a lot about human aging based on how dogs age, but it really is also to help our pets live longer, healthier lives. And if you're interested, the website is there. It's dogagingproject.com. Whoop. Oh, I pressed the wrong button. So there are lots of reasons why I think this makes sense. Thanks, Ned. So probably any of you who have dogs have recognized that dogs age very much like us. They get all of the same age-related diseases that we do. They get the same age-related declines that we do, not always at the same frequency, but they age essentially identically to people, except they do it a lot faster, right? We've all heard this idea of 7 dog years to 1 human year. It's not perfect, but it's pretty close. They've got a great genetic and phenotypic diversity, and by that I mean that everybody in this room can tell the difference between a Chihuahua and a Great Dane very easily, and that's true for almost any trait that you can imagine in dogs. Importantly, and where this really differs from the basic science work, is that dogs share our environment. So I would imagine, if you're like many of the audiences that I've talked to about this, somewhere between 40 and 50% of you have pets, and probably about 10% of you let your dog sleep in your bed. It's hard to imagine letting a laboratory animal share your environment to that extent, right? So this is really important because it now gets us out of the lab and into the real world. People love their dogs, and so this engages the public in science in a way that I think a lot of other research can't. And also, I'm a dog person. I have 3 dogs. I love my dogs. So I recognize that, again, not only is this important for what it will allow us to ultimately achieve in human aging, but if we're able to extend the healthy longevity of our pets, that's important for the pet owners and the pets themselves. So we've done a short-term study. This study was in Seattle. We call it phase 1. It's not really like a phase 1 FDA trial. But it was a 10-week trial in 24 middle-aged dogs, randomized placebo-controlled study where we treated them for 10 weeks with either the placebo or rapamycin. No significant side effects. That was the real thing we were looking for. So that was the most important outcome. But we also looked at cardiac function because, as I mentioned, we knew from the mouse work that short-term treatment with rapamycin was enough to improve measures of cardiac function, in particular left ventricular function. And so what the data here is showing is just the change in these functional measures of left ventricular output over the course of the study. And what we saw was just like in the mice, it looks very much like rapamycin is improving cardiac function in middle-aged dogs, at least within this 10-week window. This needs to be replicated. It's a small cohort, but it's very encouraging. So now we're moving on to phase 2, which will actually be held very close to here at Texas A&M Veterinary School. That will be formally enrolling dogs over the next couple of months. So if you're interested, you can nominate your dog to participate on the website. And then that will be a 1-year study, 6 months treatment, 6-month follow-up. And then what we're really gearing up for is the long-term study, a 5-year study, many more dogs around the nation to really answer the question, to what extent can rapamycin improve health span and hopefully lifespan in pet dogs. And with that, I'll stop and turn it over to Lauren, who's going to make this interactive session a little bit more interactive, hopefully.
Well, this is just such amazing science. And before we take some Q&A from you guys, I have 1 or 2 just questions. And the first thing for me when I started digging into this that really came to mind was the ethical issues, right? So, should we be trying to alter the natural aging process? Are we opening up a Pandora's box here? So whoever wants to start.
Yes, we should be trying to alter the natural aging process, and no, we're not opening up a Pandora's box.
So maybe wants to expand on that a bit.
I think that obviously we're all you kind of know where we all stand, but I sort of view targeting aging directly as preventative medicine, right? It's exactly what we should be doing if we want to give people the longest, healthiest lives possible. It's not playing God. I've heard people say that. This is no different than treating somebody's Alzheimer's disease or cancer to try to give them longer, healthier lives. It's using the same tools and technology, but I think in a smarter way and in a more efficient way.
Yeah, I think for me, it's gone. Everyone, when you think about aging, or for us, we use the word rejuvenation. All of a sudden, people start thinking, immortality or something non-scientific. But I want to echo the fact that we're using cutting-edge technology and research approaches to understand basic biology and then apply it to the human condition. And I think as long as we continue to do that and really have the right controls and attack this problem in a scientific way, then I don't think we're opening a Pandora's box. I think we're gaining a lot of knowledge that's there to be gained and having an incredible application towards our daily life.
So I guess you guys know what I think. But but to at least be sympathetic with the individuals that say that this feels inappropriate and somehow insensitive to what we are as people, a little bit of history is helpful. When the first heart transplants were conducted, which is now a routine medical procedure, there was a lot of discussion within the church as to whether or not that was okay, because that's where the soul hung out. And what you see, and we don't also believe that, thunder gods exist that control weather and stuff. And so what happens is over time, these sort of quaint ideas that constrain human innovation get put in the right box. And I think you just have to be sensitive to those that still cling to the old ideas. But, history does move on.
Okay. I think as a journalist myself, I always like to have this healthy dose of skepticism, right? It's part of why I'm here, I guess. And I hear about this science, I hear about this technology, and I think this is going to be a science for the wealthy. How do we make this accessible for everybody? Will it be? Ned, was this?
I want to take this one.
Yeah,
okay.
That one.
So I'll give you a fun story, but it's not fun for the people that suffer from it. So. End-stage renal disease, this is when your kidneys fail, which actually is what my grandfather died from. But he was also just a tremendous alcoholic and womanizer, so he had all sorts of other health problems that was, but anyway, he died in his 50s from end-stage renal disease. But in the United States, we spend $35 billion with a B, okay, treating this one disease. This is bigger than the entire NIH budget. Now, in our mice, when we eliminate senescent cells, blocking that disease is one of the 20 or more phenotypes we've seen. So what this means is that we're going to take this thing, which is consuming vast amounts of our GDP, and make it go away. So my belief is that these drugs that we're going to be creating that extend health span and eliminate these scourges that are these giant headwinds on our nation economically, these will be the cheapest drugs ever. In fact, they might be mandated in order to have certain types of insurance. Now, that's not going to happen right away, but these are going to be standard of care to make it so that you don't wind up with end-stage renal disease, atherosclerosis, osteoarthritis, osteoporosis, all of these things that cost money. And so we're going to be able to redesign the healthcare economic system around a new era in which these things can be made to go away. And I think healthcare costs are going to dramatically decrease.
Yeah, I'll just add a couple of quick thoughts. So one thing I want to say is I want to be a little bit more careful and not necessarily talk about everything going away. I think what our hope is to really push these diseases back as far as possible and keep people healthy as long as possible, because ultimately people are going to get sick and die. I mean, we're not talking about immortality. The other point I want to make, I think the insurance part is really critical, and obviously it's hard to predict where things are going to go, but this idea that these are going to be drugs for the wealthy, that will only happen if they don't become insurable. And I think, as Ned says, if it gets to the point where it's clear that these drugs are having these significant impacts from a preventative sense at keeping people healthy longer, economically, it just makes perfect sense for the insurance companies to get on board, and then they're no longer drugs for the wealthy.
Oh, just to clarify, Matt's absolutely right. What we see in our mice is that when we delay these diseases of aging, they only come at this kind of window at the end of the animal's life. So it's like they're healthy, and then they get sick and die really fast. And so you can still get these diseases, but they tend to sort of come at the end. Although many of the animals die, at least in our mice, of cancer before the onset of these diseases, but it's the idea of essentially compressing the old period to this very brief period right before you die. Okay.
One more question for me. What's that?
Lots of hands up.
Oh, all right. You know what? Then let's just get started. We only have a few minutes left. There's a microphone right in the middle there. Thank you. Enough of my questions. Let's get to yours.
Oh my goodness.
Let's go.
Okay,
Start.
good. Thank you very much, and thanks for referencing Jean Calment, when she has her famous quote, "I've only had one wrinkle in my life, and I'm sitting on it." But I agree with you, that we are right on the verge of just breaking open not just how we think about aging, but how we think about biology. And this is just a small self-interested infomercial. I hope everyone will come to my talk on the future of human genetic engineering today at 5. But my question for all of you is, as was just mentioned, opening up the funding and regulatory and insurance markets will get all of the resources to this kind of important work that we need, but there are impediments to that. And one of the, as you all know, of the impediments is how we think about aging. What are your thoughts on whether aging should be categorized as a disease? And if it should, how do we get from here to there?
I'll start, I guess. So I kind of try to avoid that question of should it be a disease or not, because I really think that's a semantic argument it's not really that, in my opinion, all that important. But I do agree that the way that we think about aging from a regulatory and policy perspective is critically important. And I think the real challenge is the idea that treating healthy older individuals, that there can be no tolerance for side effects, right? That's sort of the paradigm, is if the person's healthy, then the treatment can't have any side effects. And I think we have to get past that. I think we have to recognize that a normal 80-year-old is not the same as a healthy 20-year-old. And that at an individual level, I'm sure all of us would be willing to accept some level of risk if we could have 2 more decades of good health in our 40s, 50s, 60s. But at a policy level, how we get there, that I think is the real challenge. And I don't have the solution, except people are talking about it now. So I think progress is being made. FDA is involved in discussions in this area. So I think we will get there. But that sort of shift in the way we think about what it means to be healthy and what level of risk is acceptable for the potential benefits is the real critical question in my mind.
So I'll tack just one thing on. So what we're doing to approach this, because the FDA doesn't think of aging as a disease, and that would be so what we do is we pick very specific diseases of aging where we think we can intervene, and then we make drugs that intervene there, and we use very conventional regulatory approaches to measure the effects of those drugs and then seek approval. And so we're going to wind up getting our drugs approved on the very same endpoints that drugs that don't very well treat those diseases. And now over time, what will happen is that as you get a series of these agents approved, say for osteoarthritis or for glaucoma or for macular degeneration, over time, you'll get experience with these drugs. They're going to look safe, and people are going to start noticing off-target effects in which you get enhanced cardiovascular performance, and gradually there will become a way to have a surrogate marker to seek an approval for an aging surrogate. But if you go back and look at cholesterol or atherosclerosis and cholesterol as a surrogate marker, that took a long time. And so, but I want to emphasize that for us to make these drugs and succeed with these drugs, we don't require a change in the regulatory environment. It's totally cool for them to be as backward cavemen as they want, and we can still develop the drugs quite effectively.
So for me, really, just to quickly address that, I take it from the public perspective. You know, we can all understand treating Alzheimer's or treating cancer. However, the public and the scientists haven't met in the middle yet with respect to aging. And I actually think it's really important. And I think as more science reaches the public media in terms of, oh, look, there's a treatment, it was for aging or cognitive decline, I think that that's going to change the culture a bit. I think asking, do you treat aging as a disease is not quite right. It's more of, yes, aging can be treated or targeted. And I think changing the mindset through things like this, through things like National Institutes of Health funding more basic aging, which is all constituents that have to call, you know, the senators and congressmen. As we change that mindset, I think we're not going to have to ask that question. It's just going to be routine. Yes, of course you can treat aging.
Great, let's move on to the next question.
All right, so a great panel. Thank you very much. I'm a professor at Baylor College of Medicine. I study the ethics and policy of emerging technologies, including aging research. In other words, I study you guys who are studying aging research, formerly at Stanford and UCSF. And I have a question that actually got prompted with your introduction, Ms. Dunn. You said the quest for immortality is closer than ever before. In the same breath you said, but we're talking about living better, but not necessarily living longer. So that kind of dissonance, I think, has been set up in your talk, and if I hear it correctly, you are settling on perhaps a future that would be an extended health span rather than an extended lifespan. So if you would look forward 50 years, and by the way, Saul, old age is coming for you, I can tell you.
I'm seeing it slowly. I'm seeing it.
The gray hair, no hair, and wrinkles, also from personal experience. So if you could look ahead 50 years, what will aging look like for this group and the children of the people in the room?
So I guess I'll start. First of all, looking forward 50 years is a fool's game, right? Actually trying to predict it well. But I will say, I hope, if the science continues to progress the way that it has been, I hope that what we'll see is significantly extended health span and lifespan. I am actually in the camp that I don't think those two things are easily separable, and all of the interventions that we've talked about today seem to do both. But again, the primary concern is to maximize the period of life that is spent in good health, with, you know, free from the diseases and disabilities of aging. In terms of putting numbers on it, I mean, 50 years is a really long time, and that depends on a lot, including, you know, like what happens that's beyond any of our control. So I don't know whether it's going to be 20 years life expectancy, 40 years life expectancy increase, but I would be surprised if it's not significant increases in both life expectancy and health span are achievable. Whether that'll be true for everybody, I mean, there's too many factors that come into that.
So my stepfather died demented and incontinent in his bed at 87. And what I hope we achieve in the coming decades, but again, 50 years is like a mind-blowingly far number, I think rather than being demented and incontinent, I like the idea of dying, on the tennis court at 105 while winning, or being killed by a jealous lover at 107.
I just want a lover at 107.
I might skip that one.
Yeah. Hey, you got to go out somehow, right? So anyway, but in all seriousness, what I hope we get to observe is, I cannot accurately predict quantitatively. I think I can directionally say, with some degree of conviction, that we will sufficiently alter the duration of our health span that we will be forced to redefine the classical chapters of life. So like when you're in college, when you're in grad school, when you're supposedly an adult. A friend of mine has this term he now calls being a super adult, okay, which is like after your kids are grown up and you have to do something, and and you get to redefine who you are, a new career, perhaps an additional a degree, become a musician, if you weren't one already, and I think that that's what I hope for.
I think in thinking about that type of question, it's good to remember that we are scientists, but we're also people. And we're made up of all of our experiences, and that also influences the way that we are going to think about science and the approaches that we're going to take. And I think that's a great thing to bring to science. And I think that's what's going to cause a slightly different answer from every scientist that you ask that's doing aging research as to whether they're going to prioritize lifespan or health span or both, or a different perspective. I'm more of the camp of I'm fine if I live 90 years as long as I can read a book, go to sleep, and still think about that last chapter. But again, that's based on my experiences. So I think you really have to, when you hear these scientists saying immortality or not immortality, all of this is also dictated by our own experiences. So that's something to keep in mind, is we are scientists and we love the scientific method, but we also have our own, to a certain extent, biases as well.
Yeah, but let's not say immortality at all.
Yeah, I think that gets the field in a lot of trouble, actually, yeah.
And we don't know how to do it either.
Yeah, so let's not use that word.
Okay.
Great.
And if I can have a lover at age 107, I'll just be happy. So work on that. Okay. Next.
Great panel. I'm going to do two quick questions. One, do you think the caloric restriction or intermittent fasting would have an effect on sort of the senescent cells, basically through autophagy and so forth? Like that may be one of the main reasons why caloric restriction or intermittent fasting works. And the other thing is just keep thinking about things like resveratrol. Basically the kind of cautionary tales of things like you understand the immune system and so forth, and then suddenly it doesn't work as well, and so you think about rapamycin or NAD, those kind of things. So anyway, either one of those or both.
Why don't you let me take the resveratrol one first?
Okay,
and
I'll
then you
take
can the
so
senescent cells.
first of all, the experiment you just proposed, we haven't done yet. But my prediction, my hypothesis, would be that calorie restriction would probably reduce senescent cell induction. Because it will wind up impacting a lot of things. Okay, whether or not it would facilitate, say, immune-mediated elimination of senescent cells, we'd have to go test that. It's actually a wonderful experiment to do, but we haven't.
And so I think resveratrol is a really interesting example because there was a lot of excitement and attention several years ago about resveratrol. If you actually look at what many of us, myself being one of the primary people, were saying at the time, is that let's slow down on resveratrol. The hype has gotten way ahead of the actual science. What the difference is that if you actually look in the literature, rapamycin works. It works every time. It works well. Everybody gets it to work. Resveratrol doesn't in mice for lifespan. Okay, so I think that they're completely different ball games, and it's hard for people who aren't experts in the field to separate that. But the reality is there's science that's backed up with a lot of hard data, and there's science that sometimes gets into the mainstream that isn't. And it's a great story. I wish we could all drink tons of red wine and live longer. Resveratrol is a molecule in red wine, for people who didn't get that joke. The NAD precursors, I'll just say it because you brought it up, I think are really interesting. They're not to the level of senescent cells or rapamycin yet, but the data that's out there is compelling, and it looks really interesting. I think it's premature for people to start taking these NAD precursors just because we don't even know whether they'll do anything positive. It probably won't hurt you, probably won't help you, at least at the doses that people are taking them now. That's my take on it.
Great. I guess all that red wine I've been drinking is for nothing. Thanks.
It's not going to make you younger.
I have no more excuses. Go right ahead.
Hi, my question first is for Dr. David. I was wondering if you could comment on which are the specific biological pathways or targets that you are interdicting, and or what are the other biochemical markers, and what type of modality of drug that you are pursuing. I'm not familiar with your work, sorry. And then the second question is more of a larger ethical question, which is, I think it's key that extending the health span versus lifespan, but at some point, and this might be, sort of coldly calculating, but don't we reach a resource constraint in terms of, we'll have larger population numbers living longer, but we still have a resource constraint issue. Just something interesting to think about.
So the way we eliminate senescent cells is we started out 5 years ago, and we actually didn't know how to kill them. And so we did a big search for Achilles heels of these cells. Essentially, these are gene products that when you inhibit their function, the cell dies because it requires the function, and it requires the function uniquely, meaning a non-senescent cell doesn't need it. We don't have, and so one of the other things we learned is that not all senescent cells share the identical Achilles heel. So we have this thing called the atlas, which is a giant map of all the cells, the primary cells that we can isolate and grow in the lab on one axis, and all the molecules we found that kill senescent cells in the other, and it's like a heat map of Utah. And so what you can do is you can go in the atlas and pick your molecule out and then take it for the cell type you need to kill. And then we use relatively conventional biomarkers for senescent cells. Turns out there is no perfect one. So it's kind of like we have a list of like 7 things, and if you have 3 on the list, we say you're a senescent cell.
So are those chemicals or what?
Oh, the biomarkers?
Yeah.
They are gene products that the cells make.
Okay.
And do you want to talk about the hypermalthusian issue? Yeah. Okay.
Yeah, I guess. So the way first of all, I think that it's an important point. It gets brought up frequently. The idea that if we achieve extreme longevity, that there is going to be resource allocation issues. Like the idea that there's a reason why the older generation passes away so that the young generation can live on. I think from a practical perspective, in developed countries in particular, the kinds of potential changes in lifespan that we're talking about are unlikely to lead to significant problems from a resource allocation perspective. And I think the key thing to keep in mind is that with an increase in health span, the potential is there for increased productivity during those years. Now, what I do think is going to happen is there's going to have to be social reconsideration of what it means to be older, what's the appropriate retirement age, and things like that. There is absolutely no way the retirement age can stay where it is now if healthy life expectancy is increased by a decade or 2 decades. So I think that probably is the more important issue. That's my guess. But again, how this will actually play out, it's really hard to predict, and there will be social challenges that go along with it, no question.
And that's why health span has to be
absolutely right.
predicated
Nobody is
on
talking
any
about
lifespan.
extra
Right.
decades of frailty, right? That's what we all don't want. Yeah.
Okay. So it's 1:33. I don't know what the laws are here. We can keep going. But I'm sure these guys are happy to answer any questions after the panel. I'm just going to volunteer you guys. So thank you so much for coming. This was fascinating.
Thank you.